Great honor to introduce Dr. Lisa Barnes, who is the Alavi and Solomon Jesmer Professor of Gerontology and Geriatric Medicine and a cognitive neuropsychologist within the Rush Alzheimer's Disease Center at Rush University Medical Center. She's also the Associate Director of the Rush Alzheimer's Disease Research Center. She received her PhD from the University of Michigan in biopsychology and completed a postdoctoral fellowship in cognitive neuroscience at the University of California, Davis. She joined the faculty of Rush as an assistant professor in 1999. She has received numerous NIH grants and has published close to 300 manuscripts. And so we are really excited to hear your talk today, Dr. Barnes, and thank you again for coming.
Thank you so much. It's nice to see so many people that I know or names that I recognize. So thank you for the invitation to present. Let me share my screen here, turn on my timer. I'm going to present some of the work that we've done at the Rush Alzheimer's Disease Center to understand risk factors for Alzheimer's disease and cognitive aging in older African-American adults. And I'm happy to take questions at the end. In interest of time, I'll just try to go through. And I apologize for some of you who've heard my talk before. Maybe you'll learn something new today. I'm going to just do a few things for those of you who don't know a lot about my work. I'm going to first just discuss some of the evidence that suggests African Americans have a greater risk of cognitive impairment and Alzheimer's dementia. And then I'll describe our longitudinal studies at Rush, where we've investigated cognitive aging in older African Americans. And our studies, we also go for brain donation. And so I will talk about some of the emerging evidence coming out around racial differences in neuropathology in our cohorts. And then the last part of the talk, I will talk about some of the unique risk factors that we're looking at in African Americans. But first, I want to just give a very brief background on the problem that we're dealing with, because I understand this is a multidisciplinary audience. So Alzheimer's dementia is one of the most feared diseases of old age, and has become a public health crisis. Among the leading causes of death, it's the only one that cannot be prevented, slowed, or cured. And of course, Alzheimer's dementia is the most common form of dementia. It accounts for about 2/3 of the dementia cases in older adults. And currently more than 6 million Americans have Alzheimer's. And by 2050, this number is projected to increase to anywhere between 12 and 16 million, unless we find a cure or a way to prevent the disease. 33 to 50% of adults over the age of 85 have Alzheimer's because it's very correlated with age. And it's a very costly disease. It costs about $100 billion currently, and this number could go up to $300 billion within 30 years. And that's not even counting the unpaid informal caregiving that happens and the toll it takes on people with this disease and their loved ones. The disease is highly variable. It develops over decades and can affect a person over anywhere between 3 and 20 years. So a lot of variability in how the disease progresses. So, I study the epidemiology of Alzheimer's, and, because of that, we have to pay special attention to the changing demographics of the United States. So this slide shows data from the US Census to show you that the demographic profile of our nation is becoming increasingly diverse. And so this is showing you what the population was in 2016 and what it will be in 2050 for the different racial and ethnic groups. And what you can see is that minoritized population, so that's Asian, Hispanic, and Black people will make up the largest segment of the US population in 2050. So they'll go from 39% up to 52%. And Hispanics, Latinos will have the most dramatic increases overall. But if you were to hone in on adults over the age of 65, the population of older Black adults will double during this time period. And so this changing demographic is particularly problematic for older African Americans because it's thought that they are about 2 times more likely than white Americans to have Alzheimer's dementia. It's been documented that they're less likely to receive an official diagnosis from, healthcare practitioner or memory clinic. And they are often under-included in Alzheimer's disease and related dementia clinical trials. Although they make up 12% of the US population, they make up less than 5% of those included in trials. And, given the disproportionate burden on the population, it's just a little bit surprising that they are, so under-included in Alzheimer's clinical trials. And all of these disparities become, really more glaring when we look at these data collected by the Alzheimer's Association. We already know that there are significant barriers to inclusion in clinical research for minoritized populations. But this slide really emphasizes that a major barrier for researchers is earning the trust of people of color. So on the left, showing you the percentage of adults who believe medical research is biased against people of color. And you can see that 62% of Black adults believe medical research is biased. And on the right, we see that only about half of Black Americans believe that even when we get an Alzheimer's cure, or you could even say an Alzheimer's, treatment, only 53% believe that it will be shared equally, regardless of race, color, or ethnicity. So that is very, a sobering thing to think about when we think about moving forward and racing to a cure for this disease. So this is the disparity, using, real world data that's become a public health crisis for older adults. There's another study that just came out actually recently by Christine Yaffe and her group. But I always use this slide because it shows a broader swath of the population than what we typically see. We usually just see African Americans and whites, or sometimes we see African Americans, Latinos, and whites. But this study actually had all the different groups. And what you can see, these are data from Kaiser out there with you all. This was Northern California. And they looked at age-adjusted dementia incidence rates by race and ethnicity and over 270,000 Kaiser enrollees. And they found that dementia incidence rates were highest in African Americans, consistent with prior research, a few large studies, followed closely by American Indian and Alaska Natives. The rates were intermediate for Latinos, Pacific Islanders, and white adults, and lowest in Asian Americans. But, there's a potential challenge in interpreting results like this. Many cognitive studies demonstrate that older African Americans tend to perform at lower levels on cognitive function tests compared to older non-Latino whites. And these are the very tests that are used to make a diagnosis. But we know that cognitive function is not just measuring cognitive ability. It's influenced by many factors outside of cognition itself. And I've listed some of these on the slide: quality of education, how familiar you are with the test, early life factors. All of these things can influence your performance, and these factors vary by race and ethnicity. So when you just have a one-time measurement, which we often have in these studies, it really presents challenges for interpreting the racial differences, especially if you don't take these factors into account. So because most studies of racial differences in cognitive aging have actually been cross-sectional, we've tried to get around this challenge of interpreting the racial differences by using longitudinal designs, in which you examine a person repeatedly over time. So this is really a strong approach in studying health disparities because it allows you to gain a reliable estimate of change in cognitive function over time. And since aging is fundamentally a process of change, it makes sense to use a study design where you're actually measuring the aging process. Another important design feature is that with this design, we're able to control and hold constant the level of function that a person starts at. And each person serves as his or her own control. So you're measuring how a person changes over time based on where they started, rather than comparing people that might vary on any number of those characteristics I showed in the prior slide. So with using this type of design, we aim to identify risk factors, either genetic or environmental, and learn how the risk factors associate with cognition over time so that we can develop strategies to prevent Alzheimer's dementia, either by modifying lifestyle behaviors or identifying druggable targets to develop medications. So we have a number of cohort studies at Rush that allow us to, to do these types of things. And I'm going to just first describe the studies, and then I will present some of the data. So the Minority Aging Research Study, known as MARS, is my study, and I started this study in August 2004. And currently it consists of about 800 African Americans who enroll. They have to be at least 65 years of age, self-identify as African American, and they cannot have dementia when they start, or known dementia, no diagnosis. We recruit from the community. We go to churches, senior buildings, and organizations in Chicago that cater to older African Americans. And once enrolled, all people agree to have annual in-home cognitive testing. So we go to them, they never come to Rush, and we measure a host of risk factors, and we, take their blood in the home. Our follow-up rate has been very good, over 90% among survivors. And I was refunded, I think last year, for years 16 to 20. So it's been refunded now 4 times. Over time, about 10% have developed Alzheimer's dementia. And we have about 140 autopsies to date. And this is really merging our data across several cohorts, because it's really difficult to get autopsies from African Americans in a single study. We use a community-centered recruitment and engagement approach, in which our philosophy is in the heart of the community, ever seeking to win the community's heart, to really emphasize that recruitment into these types of studies, especially with diverse populations, requires significant relationship building and giving first before we ask for anything. So we are always out there before we even present research. We're trying to, build relationships and build trust. And because we want to keep people in the study for many, many years, until death, we have to spend quite a bit of effort and resources in retention. And so we try to build culturally celebratory retention events that keep people invested, interested, and engaged in the research. So just to give you a snapshot of what the participants look like, there's about 800, as I said. It's predominantly women, which is, consistent with lots of aging studies. Women tend to volunteer, for research more than men. They live longer. The current mean age is 84, and the mean education is 14. So they're, relatively educated, which is also consistent with the healthy volunteer cohort. Over time, we've had over 200 die, and we also have an MRI study embedded within MARS. And so we have had over 370 receive an MRI. Some have gotten multiple MRIs over time. And a little bit more than half of those alive and active have agreed to give their brain at the time of death. But of course, there's all kinds of challenges in actually having that manifest. And maybe we can talk about that later if there's time or interest. Now, MARS is completely African American, which allows us to understand within race heterogeneity and, race relevant risk factors. But sometimes you want to ask questions about, between racial differences questions. And so for that, I have the benefit of having access to 2 other cohort studies of aging and Alzheimer's that have been going on for more than 20 years at Rush, called the Religious Orders Study and the Rush Memory and Aging Project, which we now call RADC. And these 2 studies together consist of over 3,000 older people without known dementia, and they're recruited from across the US. The cohorts are predominantly white, about 91% white. But like MARS, they all have agreed to annual detailed clinical evaluation, risk factor assessment, and they give blood. But unlike MARS, both of these studies, people have to agree to organ donation at death in order to be enrolled in the study. So it's a requirement for the study, something you cannot do in diverse populations. In MARS, organ donation is optional. And so because of this requirement in RADC, they have over 1,200 autopsies. So, we've learned so much from these 2 studies because of the brain tissue from these really altruistic participants. Importantly, there's significant overlap in risk factors between MARS, and we all use the same cognitive battery, which allows us to merge the data. So all three studies use the same longitudinal design, which I'm showing here. And it allows us to use a single database to examine risk factors that are measured at the baseline evaluation. Let me get my pointer. So we measure in people who don't have any disease at baseline, we measure their risk factors, and then we follow them annually over time, assessing their cognition every year. And over time, some people are going to develop Alzheimer's disease, some will develop mild cognitive impairment, and a lot will remain cognitively unimpaired, but we're able to relate the risk factors that we measured at baseline to change in cognitive function, to incident disease. And we have autopsy built into this design so that when people die, we can measure a range of pathology in the brain from no pathology all the way to more severe, and relate those same risk factors to the underlying pathology. But importantly, we can try to understand how or if biology might be a mediating factor between the risk factors and disease. So all of the studies use the same cognitive function battery, just traditional neuropsych tests. We have about 19 tests, but we are able to measure 5 different domains, including episodic memory, semantic memory, working memory, perceptual speed, and visual spatial ability. So we've been following people for a long time now. And so, following them, measuring their cognition every year. And so here I'm just showing you a spaghetti plot of about 150 participants, random participants that we selected. And just to orient you to the slide, we've plotted age on the horizontal axis and the global cognitive function score on the y-axis. And so higher scores are better cognitive function and negative scores are worse cognitive function. The black lines represent the Black adults and the red lines are the white adults. And what you can see is that there's an incredible amount of heterogeneity. So each line is a person and it shows you their trajectory. And you can just see that some people start out high and they kind of maintain their cognition. Others start out lower and they really decline quickly. And you have all kinds of patterns in between, right? But the point is that you have these wide individual differences in where people start and how fast they decline. And that's true in both African Americans and white adults. But in the mixed effects model, when we control for demographics, we get a pattern shown in these two graphs. So here I've divided the baseline age range into 5-year intervals and plotted the predicted 5-year passive decline as a function of the Z-scored composite cognition score. So the top panel shows trajectories for semantic memory and the bottom panel shows trajectories for perceptual speed. Whites are represented by the green lines on top and African Americans by the blue lines in each graph. And what you can see is that there are level differences in cognition, with African Americans performing more poorly at every age, but the rates of change shown by the slopes of the lines are mostly similar, suggesting no racial differences in rates of change over time. The only difference we're seeing is really in level, where people start. Now, if you look out past 85 to 90, you see that it looks like maybe whites are declining a bit faster. The slopes are steeper, but the data are so sparse out there. We have, not that many people, especially African Americans, living to those older ages. So we're a bit more cautious in interpreting those data. But, anywhere between 65 and 80, it really looks like there's no difference in how people change over time. We've replicated this result in our population-based study that we have at Rush called the Chicago Health and Aging Project. This is like a population of over 10,000 people, Blacks and whites, who are recruited from 4 contiguous neighborhoods in Chicago. And, we give them a brief battery of tests. And so here I'm showing you data of people who started at age 68, who started at 73, or who started at 78. And you can see the same pattern. Whites start out at a higher level on our cognitive function test, but the lines are parallel. No matter what age you start, there seems to be no difference in how people are changing. Even in people with dementia, we see a very similar pattern. So these are data from 2 different studies, 2 different populations of people who all had a diagnosis of dementia. And so on the left, African Americans are the dotted lines, and on the right, they're actually the solid lines, 2 separate papers, but you see the same pattern. African Americans start off lower, but they look like they decline actually slower over time. Everyone in this study had dementia. So far I've showed you that there's, increased risk of Alzheimer's among African Americans, which is the case when we look at these large databases where people are looking at the diagnosis. But in our data, we only see significant level differences in test performance, and we don't really see any differences in how people are changing over time. And this is whether we look at, people in our volunteer cohort, people in the population, people, who have a diagnosis of clinical Alzheimer's, we see the exact same pattern, level differences, but no differences in change. So our next question then was, are there racial differences in the underlying brain pathology associated with Alzheimer's dementia? And that's another way to sort of tackle this question about, this racial disparity. And so in our studies, as I mentioned, we follow people over time and then we get their brain at the end when they die. And so here I'm showing you two images of the brain, one with neuropathologically defined Alzheimer's disease and one without. And you don't have to be a neuropathologist. You can see that these brains clearly differ. And so, we try to measure the underlying pathology when the person dies, not we, our neuropathologists. And so we know that beta amyloid and tau neurofibrillary tangles define the hallmark Alzheimer's disease neuropathologic changes. Yet people with Alzheimer's dementia, the clinical symptoms, they often exhibit postmortem evidence of other neuropathologies, including, Lewy body disease, TDP-43 pathology, hippocampal sclerosis, as well as vascular changes, things we can see with the naked eye, like macroinfarcts, but also things we only can see under the microscope, microinfarcts, cerebral angeloid angiopathy, and large and small vessel disease. And in our studies of over 1,300 brain autopsies in white adults, we've shown in numerous studies now that Alzheimer's dementia is pathologically heterogeneous and biologically multi-layered disease, with most people having a mixture of different pathologies. So most people who die with Alzheimer's, people who come to autopsy, they have a mixed picture of different pathologies when they die, close to 60%. But what do we know about this in African Americans who are grossly underrepresented in autopsy studies? Well, we looked at racial differences in the underlying pathology of people diagnosed with clinical dementia in patients that were recruited from our memory clinic. And so, everyone comes to the clinic, because they have Alzheimer's or they suspect they have Alzheimer's. And then when they die, the family naturally wants an autopsy so that they can get a definitive diagnosis. And so when we looked at these brains of people who died and, gave us their brain from the clinic, we found that African Americans were much more likely to have mixed pathology as a cause of their dementia. And I'm showing it with the red pie slice here. So, we looked at all the different types of pathologies, and mixed pathology was much more prevalent in African Americans than it was in whites, who were much more likely to have Alzheimer's disease and, mixed with other things in their brain. But as I said, all participants in this study were recruited from a memory clinic. And we know from other studies that African Americans often present to memory clinics either later or, not at all. And in fact, when we looked at the mixed pathologies of our African Americans, we found that the types of pathologies that in that group of mixed pathologies was, Alzheimer's changes mixed also with Lewy bodies. And we know that Lewy bodies are associated with, behavioral problems, hallucinations, sleep disorders, things like that. And so it suggests that maybe it's a potential bias, selection bias, and who shows up to a memory clinic for, for different reasons. So perhaps African Americans, when they come to a memory clinic, they're not coming because they have memory problems. Maybe they're coming because there's behavioral problems that are really, interfering with the caregiver being able to take care of them. And so they come because of that. And, other studies are starting to show that as well. But, in our studies from the community, where we recruit people who don't have dementia, we're seeing a very different pattern. So in the first 89 people who came to autopsy in our community studies, we compared them to about 89 whites who were matched on key demographics. And I'm just showing you some of the differences clinically that we saw. African Americans were more likely to have blood pressure, higher blood pressures, hypertension, smoking history, and they were much more likely to have an E4 allele, which is the genetic risk marker for Alzheimer's. But when we looked at the actual pathology under the microscope, we didn't see any of those racial differences that we saw in our clinic sample. The only thing we saw was that African Americans were slightly more likely to have infarcts, mainly gross infarcts, the clinical stroke, but underneath the microscope is called an infarct. But when we adjusted for vascular risk factors like hypertension, diabetes, the racial difference went away. So, we see these racial differences in pathology in those who present to a memory clinic. Maybe it's a selection bias on who shows up to a clinic. But, when you recruit from the community and people start out without dementia, you follow them over time and they die, we don't see any racial differences in the underlying pathology in those people. So then our question was, maybe we need to focus on risk factors that might help us understand the level differences that we're seeing in cognition. And so, we and others have published numerous studies, documenting a lot of these different risk factors. These are sort of the established risk factors for Alzheimer's. And there's some that increase your risk, and some that decrease your risk. And most of these are non-modifiable. But, we're interested, in potentially modifiable risk factors at the Rush Alzheimer's Disease Center. And we have published a number of different factors that seem to increase your risk, some that seem to decrease your risk. And some of these, appear to operate the same across race. And we don't have, as much data in African Americans as we have in whites. And so, that data is slowly accumulating, but there's, some factors that are protective, and some that increase your risk, and those appear to operate the same regardless of race or ethnicity. But there are other risk factors that appear to show race differences. And, there's some that decrease your risk. And I have, W means that they decrease the risk more so in whites than in Blacks. And then there's some that increase your risk. And so far, we've only found ones that increase your risk for white adults. But I want to just spend the rest of the time talking about some of the risk factors that we have been particularly interested in looking at our African American participants. Now, one is the ApoE allele. We know that's the strongest genetic risk factor for Alzheimer's, but numerous studies have shown that it doesn't operate the same across race. Although African Americans are much more likely to carry an E4 allele. So, there's 3 different alleles. E3 is neutral, E4 is a risk gene, and E2 is the protective one. So African Americans are much more likely to carry the E4, but it usually has a weaker or sometimes non-existent effect on cognitive decline in African Americans. And so we and others have shown that E4 tends to have a selective effect on episodic memory. And so we hypothesized in this study that maybe the literature is so mixed with E4 in African Americans because, most studies that had looked at it were only really looking at using a brief screening measure like the MMSE and, maybe mask or missing any specific associations with different cognitive domains. And so we thought if we looked at the different domains of function, we might, be able to pick up something that was going on. And so as shown in this figure here, we did see that African Americans who had at least one E4 allele, they had a faster rate of decline in episodic memory, shown here. And this was true for whites too. So whites are the longer line because they have been followed longer, but you can see people who had an E4 had a much faster rate of decline. In contrast, we did not see this for any other domain. So this is just working memory as an example. You can see that whether or not you had an E4, you had the same rate of decline if you were African American, but whites had a faster rate of decline if they had an E4. And they had that, whites showed that pattern in every domain, whereas African Americans only showed it in episodic memory. So that is one potential reason why the literature is so mixed. There's another potential reason that we wanted to explore, and that is there's a gene that's right next to ApoE. on chromosome 19 called TOM40. And most genetic studies include only whites. And so in white adults or white people, the TOM40 gene is in linkage disequilibrium with E4. And so it's right on top of each other. And so it's been thought that this gene did not have an independent effect beyond E4. But if you look at the cluster of TOM40 haplotypes, so we looked at this in our cohorts, and what I'm showing you here is we see the same pattern. If you have an E3, you have the same pattern of this TOM40 gene. But on the E4 allele, there's a different pattern. So for white people, there's two clusters here in the long range. But in African Americans, they have a third cluster here in the short range. And so the question becomes for us, does this heterogeneity in these clusters, could that help us understand why E4 is having a different impact across race? And so the answer seems to be yes. So this study, we looked at the risk of Alzheimer's in people who had the E4 or the TOM40 long gene. And so, I'm showing you data on in this panel here. And as I said, E4 and TOM40 are highly concordant in white people. And so you see an almost identical effect size and effect pattern for E4 and the 523 long allele for TOM40, shown by these two red lines. The pattern is very different for African Americans, and I couldn't even put them on the same slide because I needed to show you how what the patterns look like. So in the middle pattern, we see there's a strong effect for Black Americans who have the E4 related to a higher risk of Alzheimer's. But we don't see, if you look at the 523, we don't see that same effect as we saw in whites. So it's possible that the effect of this adjacent gene in African Americans explains the mixed results in the literature for E4. Because we know that African Americans in the US are a highly admixed population. And so it's, some people have the cluster, similar to white people, and some have clusters similar to Africans in Africa. And so, perhaps this is explaining some of the mixed literature that we're seeing. And we, extended this study to look at cognitive decline. And this was led by Casey Dieters, who's now at UCLA. She looked at TOM40 with cognitive decline in our cohort, in the same cohort I just showed you. And, I'm just reminding you about the mixed, this extra cluster that we have here for Blacks. So here whites have the 2 clusters and Blacks have the 3 clusters. And what we found, here is decline over time. So whites, who have an E4, their TOM40 is, right on top of each other. So it's just one line. But because Black adults have either the short or the long TOM40, we saw that it was actually the short that was protective in African Americans. So people who had either one short allele or 2 short alleles, they actually had a slower rate of decline than those who had the long allele. So again, showing us that there's some heterogeneity, and I think the take home message is, that, when we're looking at these cohorts, if we only have white people in our studies, we are remaining blind to these types of differences that might be in our data. And we're making assumptions on the disease based on one population. So, that's a genetic risk marker, and it seems to operate differently across race, but you really can't do anything about your genes. But we also know that race is a social political construct that's rooted in cultural identity and shaped by historic and current events. And so in our diverse cohorts, we have been documenting a number of culturally relevant risk factors. Some, have been called out in the recent NIA Health Disparities Research Framework. Others we took from public health and social epi studies. And I'm only going to focus on the green ones for now, in the interest of time. So one of the risk factors that we've been interested in is trying to understand the historical factors that we think might have shaped the lives of older African Americans. And so from 1916 to about 1970, there was something called the Great Migration, in which about 6 million Black Southerners relocated to urban areas in the North and the West to escape the toxic Jim Crow South. And in one of our studies, we examined the effect of geographic place of birth on late life cognition. So all of our participants live in Chicago now, but about 40% migrated at some point in their lives to Chicago. And so, we asked them where were they born and where were they living at age 12, a time when most people would have been in elementary or high school. And so consistent with previous studies that have shown adverse health effects for Southern US birth, we found a similar pattern. So those people who were born in the South or living in the South at age 12 perform more poorly on our global measure of cognition. So lower scores are bad for you compared to those who were born in the North or living in the North at age 12. But interestingly, this exposure was not related to change over time. It only was associated with where you start, your level effect. And so, there's lots of things that are different between the North and the South, especially during that time. But one thing that we know was different was the educational experience of Black and white children during that time. People in this age cohort, they would have been coming of age right before the Brown versus Board of Education decision. And so the cohorts that we're studying now, they actually represent the first generation of Black adults to attend, or Black children to attend legally desegregated schools after that decision. And so we asked people in our cohorts, did you attend a segregated school, and how many years and things like that? And we didn't find that segregation experience itself was associated with cognition. It wasn't a main effect, but we did see an interaction such that Black adults who reported being born in the South or going to school in the South and attending a legally desegregated school had the worst performance on our cognitive function tests, even compared to their counterparts who were also born in the South and going to school in the South and attending a legally segregated school. And you can see the performance for those in the North performing a bit higher. And so we were surprised by this result because, everything that I'd read, suggested that the segregated schools would, had less resources, maybe the teachers were not as prepared, all kinds of differences between, the Black schools and the white schools, how much money was spent on schools. But, we hypothesized that maybe there was something about attending a school where, you're not wanted. We've all seen the pictures of, Ruby Bridges being escorted with the guards, people throwing and, shouting, really terrible things at these children. Maybe that was a toxic experience, and now 50, 60 years later, when they're in our study and we're measuring their cognition, we are able to see that, this accounts for some of the performance, the lower performance in our African Americans. So just a hypothesis, but one that we think is really interesting. Another persistent lived experience for African Americans is discrimination. Discrimination has been shown to be a risk factor for poor mental and physical health in numerous studies for over 2 decades, but it's been mostly studied in young and middle-aged adults. And some, but not all, studies have found that it partially explains disparities in health. And we were particularly interested in this risk factor given the historical nature of discrimination for older African Americans who would have come of age, and lived through Jim Crow South. And so we use the Williams Everyday Discrimination Scale to assess exposure to everyday occurrences of unfair treatment. And these are some of the example items on that scale. And you can see that the frequency was rated on a 4-point scale. How often do these things happen to you, from never to often? And following previous work with this measure, we recoded the responses to a binary format, and we summed across the items to get a total score that ranged from 0 to 9. And in fact, we've published a number of studies looking at different outcomes, from quality of life to blood pressure to depression and even mortality. And we see this, that perceived discrimination is a potent risk factor for all of these outcomes. But we were one of the first groups to look at this exposure with cognition in older adults. And so using this scale, we found that higher reports of discrimination were associated with poorer performance on cognitive function, particularly episodic memory and perceptual speed tests. In a separate study, we found that people reporting more discrimination also had higher levels of C-reactive protein in their blood in a dose-response fashion. So people who reported more discrimination had higher levels of CRP, and we know that that's a marker of inflammation. And so that could be one potential biologic mechanism linking discrimination to poor cognition. In a recent fMRI study, we showed that higher levels of discrimination, those people who reported higher levels of discrimination also had this differential functional connectivity in their brains in areas that are involved in trust perception. So we found a differential connectivity in the insula connected to the dorsolateral prefrontal cortex, suggesting perhaps a neurobiologic underpinning of discrimination. And in a recent study that I'm trying to finish writing up, we found in 76 Black adults who came to autopsy that higher levels of discrimination are also associated with the higher odds of atherosclerosis and late, which is TDP-43 with or without hippocampal sclerosis. And these associations were independent of having vascular risk factors or depressive symptoms. But we don't have to only look 60 years back or look at early life to examine the adverse effects of discrimination and racism. The structural racism within our society has been amplified by all the recent events that have happened linked to police brutality, COVID-19, the persistent poverty that we see for Black and brown people. And so, we've tried to understand how living in a society where you are consistently marginalized, how that might have an impact on aging. I mean, we use a general marker of stress called the perceived stress scale, of the Cohen 4-item scale. And so this scale captures the degree to which a person appraises their life as unpredictable, uncontrollable, and overloading. We also, in a separate study, looked at the correlates of perceived stress in our older Black adults, and we found things that you would, naturally think are stressful. So, having more neighborhood problems or financial burden or being socially isolated, all those things that I have listed on the slide appear to be associated with higher levels of perceived stress. But when we measure perceived stress in our study and measure cognition, we found that those people who were reporting perceived stress at the 90th percentile of the scale had a faster rate of cognitive decline compared to those who reported perceived stress at the 10th percentile. So perceived stress does appear to be, could explain a lot of these risk factors that we're seeing, these markers of race-relevant risk factors with change in cognitive function. And these effects for perceived stress were particularly pronounced for episodic memory and visual spatial ability. The last risk factor that I'll show you is a social construct that we also measure in our study called John Henryism, which is a strategy for coping with prolonged exposure to stressors like discrimination. This was developed by Sherman James, a noted epidemiologist and race scholar. And he developed the scale primarily to look at how it related to cardiovascular disease in Black men. So we wanted to see if we could see an association with cognition. And in fact, we did find that people who reported more John Henryism, they're more likely to use, the John Henryism way of coping. They perform worse on our cognitive function test, but there's no association with change over time. So it's really only affecting level. And we found that higher reports of John Henryism was associated with lower cognition, an effect that was similar in impact to having fewer years of education. So I know I've told you a lot in this study. I've tried to paint a picture. We have this cohort that we've been following over time where we can do all kinds of things. We can measure their cognition. We can measure risk factors and relate them to cognition. We can get the brains and measure the risk factors to the pathology. And I think the picture that's starting to be painted, at least for me, is that, we have this persistent racial disparity in Alzheimer's dementia. but it seems like it's probably due to racial differences in cognitive level where people start off, right? So we're using these tests. I mean, I know the diagnosis is much more than neuropsychological tests, but that's a big part of how we make the diagnosis. And, the starting level we're seeing is really highly influenced by the context in which one lives and grows up and needs to be taken into account. There's a growing body of literature, longitudinal studies showing in different cohorts, not just in our volunteer, but also a population. And it's been shown in HRS that there's really no racial differences in cognitive decline over time. And there's a smaller but growing body of studies that suggests that the underlying neurobiology of cognition is not different, if you recruit from the community. Because there are these level differences, we need to know why, and we need to understand, what are some of the factors. And so there's a number of race-relevant risk factors that appear to be associated with level of cognition. And these could set the stage for future intervention and policy work to help us eliminate disparities. And I think moving forward, it's going to be really important for us to start to design studies that can incorporate the life course and the lived experience of our diverse participants, because that's going to help us advance the science of disparities and ultimately move us towards health equity. I'd just like to acknowledge the funders for the work that I've presented and our participants of all of our cohorts at Rush, because without them, none of this would be possible, obviously. And all of the faculty and strategic partners that we have at Rush and the staff who collect all the data. This is really a team science effort, and nothing can be done with just one person or just one cohort. So I'm really fortunate to be able to work with such smart people. And so this is our research resource sharing hub. If there's anything that interests you or you want to see what kind of data we collect, we have everything: cognition, omics, everything, brain tissue, and we share widely. And so you can go to that website, or if you have questions and you want to, collaborate, whatever, it's my email. And we have supported many young scholars and case scholars, and people who want to put in Alzheimer's Association grants. So happy to work with anybody. So thank you for your attention. And I will stop sharing so I can see you.
Thank you so much. That was really a fantastic talk, and we appreciate you giving that talk. So I'll just manage the question and answer session. Hector, I saw your hand up. Did you have a question?
Yes, thank you very much. And Lisa, such a pleasure to see
Hi,
you.
Hector.
Thank you for coming. How you doing?
Good.
Wonderful talk. Thanks for sharing this, what you've been doing for the last few years, let's just say.
Yeah, I started in high school.
Yeah, it's been a while, right? So the pathology, I was really fascinated, and I was struck by the higher number of infarcts in AD you're seeing among African American Blacks compared to whites. But paradoxically, it seemed like the atherosclerosis and arteriolosclerosis was lower compared to whites. And that just doesn't
make sense, right?
Right. So it doesn't make sense, but this will maybe a question you can, or what's going on there? What do you think?
I don't know, honestly, Hector. That has puzzled me as well. It could be how we're measuring small and large vessel disease in the areas that we're, measuring, we're just missing some of it. It could be even a selection bias of people who show up, to get autopsy. I don't know. I think we need more numbers to be able to tease all of that out, because we certainly don't see that discrepancy in our white sample, but we have over 1,300, autopsies. So we probably need more data.
Probably so, maybe, and nothing on the MRI that would support that. Nothing, huh? Okay.
Nope.
One of the things you're talking about, Sherman James's work on John Henry is really fascinating, and one of the things that Sherman was apt to talk about is the maladaptive coping mechanism there. That is more John Henryism. You're facing this adversity, just like the legend of John Henry. For those of you that remember that, in the face of so much adversity, you just keep trying until you exhaust yourself. And it's associated with cardiovascular disease, John Henryism. Higher John Henryism means increased risk for MI, CHD, and other bad cardiac events. And to me, it seems very intuitive that increased John Henryism would also be associated with, infarcts, as well as some of your athero and arteriolosclerosis. So have
Yeah.
you looked at that, or?
We have not looked at that yet. We started with cognition just to see if we could get a signal. And we
Yeah.
saw that, associated with lower cognition, but not change over time, which
Yeah.
suggests to me that we won't see anything with pathology since it doesn't seem like it's affecting the disease. It's just
Mhm.
affecting where you start, but we haven't looked at it yet.
Yeah, I would imagine it would operate through like a vascular pathway, but
Probably.
more to do.
Yeah.
Anyway, loved your talk. Good to see you.
Thank you.
Linda.
Yeah, thank you very much. That was a wonderful talk, a really nice overview of so many areas. It's so multifaceted, and we really need to do so much more research here. I was wondering about somewhat of a disconnect between the fact that you were saying that in your community sample, you're not seeing a lot of differences in the neuropathology and the people come in based on the community. But there seems to be some differences in the genetic underlyings, like the TOM40 and the ApoE4. And so how is the genetics informing the pathology, and what kind of differences might we expect to see because of these differences in the genetics?
That's a great question. We really haven't started to look at that. I mean, it takes so much time and effort to get the autopsies.
Yeah.
And, when I started at Rush, I started to learn the autopsy protocol and literature by looking at gender differences as I was waiting to accumulate brains. And, you really need about 150 to really be able to do these kinds of studies, which I don't have that number yet. So I'm not really sure what would happen with the genetics. I mean, we know that ApoE is highly associated with amyloid in whites, but it should be the same in African Americans, right? Because there's really no biologic difference between the races. But I don't know. It's an open question. And maybe looking at TOM40 will give us some more answers. I mean, to me, that was the most exciting thing about ApoE, seeing that there's this TOM40 gene, and people, they still don't really believe it because most studies have been done in whites. It was really, a little challenging to get that published because people are like, TOM40 is not doing anything. But when you have the samples and you can actually look across race, you see that there is this different pattern. So that's something we'll be doing as we accumulate more brains to see the genetic effects. Thank you for that question.
Judy.
Thanks, Dr. Barnes. That was such a wonderful talk. It's always a pleasure to hear you speak, and I learned something new every time.
Oh, thank you.
I just wanted to mention with the TOM40 finding, it's so interesting, and I really hope you and Casey continue to follow it. But that TOM40 gene has a big influence on mitochondrial function, and so it's a mitochondrial gene. And just thinking about the risk factors that you're seeing in your African-American participants and what others are seeing as well, I do wonder if it's possible to think more along those lines from a metabolic disorder and approach specific to TOM40, because that is its function. And if you ever want to dig into it more, there's this whole history around TOM40 and ApoE4 and some secret conspiracy theories with it.
Really?
Because I'm a part of a mitochondrial Alzheimer's working group. And just as you say, how they're in linkage disequilibrium, that there's a specific form, the long form of TOM40 that perfectly overlaps with ApoE4.
Mm-hmm.
And so the ApoE4 gene was discovered a year before the TOM40 gene as a risk factor, and therefore ApoE4 got all of the attention. But there's this entire other field that's working on TOM40. And because you and Casey and team are really seeing this with TOM40, it'd be interesting to try to chase it down a little bit more biologically and through the functional pathways, because there is a lot known about TOM40 and its function outside of the Alzheimer's world.
Oh my God, thank you for that. I would love to learn more about that. Maybe I can contact you offline to get just some of those papers. I had no idea.
Yeah, it's really interesting.
Yeah. And we can look at those. We can look at the mitochondrial differences, So that'll be interesting.
Mm-hmm. Yeah.
Thank you, Judy.
Cheryl.
Yeah, thank you so much, Dr. Barnes. I don't know if you can see the chat, but
No,
you're getting Yeah, so a lot of excitement about, some of the links you've made between displacement and cognition, how meaningful your research program is. Some people would like to have maybe a copy of your slides if possible. They're absolutely fascinated by the work that you're doing. So I have a question really that is going to maybe extend what you've presented a little bit, because you struck me in the beginning of the talk by talking about how urgent and devastating the problem of Alzheimer's is, particularly for any demographic group, but particularly for the African American community. And I noticed the etiologic associations that you presented throughout. Some were had pharmacologic implications, other had non-pharmacologic implications. And much of it, in addition to talking about individual level issues, were structural, right? And so the question is, given the devastation of this public health condition, where do we start to intervene? What's the landscape right now around interventions? Maybe, just kind of position for us the top pharmacologic or non-pharmacologic intervention targets, because, I find this so distressing.
Yeah, you're right. It is very distressing. And unfortunately, the pharmacologic interventions, I can't really speak to. I know that, as I said, African Americans are under included in those kind of trials. And, in fact, the Engagemen trial that got so much press, there was maybe 3 African Americans in that trial. And this was a drug that was, fast tracked to FDA. Now that's all changed. But so that's a huge problem. But there are a number of non-pharmacologic interventions that people are looking at now, and I know that they're trying to become much more diverse. So, for instance, the Pointer study is going on now, which is a multimodal intervention that's looking at diet, physical activity, and cognitive activity to see if that might slow cognitive decline in a wide range of people, right? So diet is going to be one place where we could definitely intervene. We just finished a trial, the MIND trial, which I can't tell you the results yet. We didn't have very many African Americans in that trial, unfortunately, but Pointer is going to improve on that and, have much more diversity in their trial. I think another area where we could probably intervene is cognitive activity. There haven't been that many trials. There've been some cognitive training trials. It doesn't really show that the effects persist over time, but there's got to be ways that we can be more innovative and get more people enrolled in these kinds of studies, because that's something you can change, right? How cognitively active you are. There are other factors that we found in our observational studies that you cannot do an intervention for, like social activity. You cannot, really intervene on something like that. So we'll have to think about where our top priorities are, things we can actually change. So I would say I put my money on diet, sleep, and cognitive activities as places where we can intervene, because you're right, a lot of this is structural and systemic. And in fact, in my renewal, I'm measuring some of those systemic markers, like in neighborhood factors that might help us understand. It's much harder to change those things, right? It really takes policy changes. But if we can start maybe at the individual level and maybe multimodal style, we might be able to move the needle a little bit. But it's a really tough problem.
There's the optimist in me says, the things that you described are targets for, other organs like the heart and, and for chronic conditions like cancers. And so maybe we need to, be more mindful about pooling our efforts and how we lay out these interventions so that we can look at collateral benefits.
Exactly. Great point.
So we are at the top of the hour. Is 12:01 or two for you. So we just want to thank you again for giving this wonderful talk, which, as you can tell, was very well received. And we hope to invite you again in the future, Dr. Barnes. Thank you again.
Thank you so much. It was a pleasure being with all of you today. Happy
Thanks,
Thanksgiving.
Dr. Barnes. Take care.
Happy Thanksgiving.
Bye-bye.
Bye.